everyone. I'm Whitney Espick, CEO of the MIT Alumni Association, and welcome to the MIT Alumni Forum. In recent years, conversation about brain health have moved out of the clinic and into everyday life. As populations age worldwide, more families are confronting Alzheimer's disease and other forms of dementia, and sometimes sooner than expected. At the same time, we're seeing an explosion of public attention around the brain, from new AI systems inspired by neural networks to growing awareness of how sleep, rhythm, and environment shape cognition, to headlines about potential breakthroughs in treating neurodegenerative disease. These developments remind us that understanding the brain isn't just a scientific challenge. It's a societal one, touching public health, caregiving, technology, and how we think about aging. Today's conversation brings us to the frontiers of that challenge, how the brain's cells, circuits, and rhythms work together to sustain memory, and what happens when those systems break down. To help us explore this, we're joined by one of the world's leading neuroscientists, Professor Li-Huei Tsai. Professor Tsai holds several key leadership academic roles at MIT. She is the Picower Professor of Neuroscience at MIT, Director of the Picower Institute of Learning and Memory, Co-Director of the Alana Down Syndrome Center, Professor of the Department of Brain and Cognitive Sciences, and a Senior Associate Member of the Broad Institute. Her work is reshaping how scientists understand Alzheimer's disease, expanding the field beyond individual molecules to brain-wide networks, and revealing how restoring communication between regions of the brain may help protect memory and cognition. Moderating today's discussion is Pam Bellick, an MIT Knight Science Journalism Fellow and a Pulitzer Prize-winning health and science reporter at the New York Times. Pam has spent her career translating complex medical and scientific advances into stories, providing clarity, context, and awareness to important public health issues. If you already have questions for today's discussion, please add them to the chat box below and upvote the ones you find most interesting. Closed captioning is also available, which you can access by selecting the CC icon in the lower right corner of the video player. And now, it's my pleasure to turn things over to our featured speaker, Professor Li-Huei Tsai.
Thank you, Whitney, and the Alumni Association for having me here today. So it is a tremendous pleasure. So today I'm extremely happy to present to you our recent progress on our effort in restoring aging brain health in Alzheimer's disease. So this is my disclosure information. I'm a scientific co-founder and SAB member of two MIT spin-off companies, Cognito Therapeutics and TAC Therapeutics. So as Whitney mentioned earlier, that my lab has been using network-level approaches to study neurological diseases and dementias. So we study the diseases using diverse experimental approaches and systems. At one level, we look at the brain circuits and neuronal population dynamics to understand the vulnerable circuitry in the brain and trying to figure out how we can leverage the intrinsic brain neuronal firing properties to restore homeostasis of the brain. And at the second level of approach, we have been doing multi-omic analysis of human samples, including brain samples and blood samples, cerebral spinal fluid samples, and also using single-cell genomic methodologies to try to understand really what happens in the individuals living with diseases, and using artificial intelligence and machine learning pipelines to gain further insight to identify new targets for intervention. And finally, we model the disease, not just using animal models, but now we actually rely heavily on human patient-derived stem cells to model the disease, and in particular, to learn the functions and pathological processes associated with major disease genes. So today, in the interest of time, I probably will only be able to cover the first two approaches in my lab. So first, let me tell you a little bit about dementia. What is dementia? Dementia is an umbrella term for loss of memory and other thinking abilities severe enough to interfere with daily life. And there are many different types of dementias, but Alzheimer's disease is the most common type of dementia, representing anywhere between 60 to 80% of cases. But you probably have also heard that there's other type of dementia, including vascular dementia, Lewy body dementia, frontotemporal dementia, and all of the dementias represent different disease presentations and different pathology in the brain. So when it comes to Alzheimer's disease, the disease was first described by the German physician Alois Alzheimer back in 1906. So this was more than a century ago. And back then, Dr. Alzheimer, by looking at the postmortem brain samples of his patients, he already identified the key pathological features of Alzheimer's disease. So these two pictures I presented here today actually are hand drawings by Dr. Alzheimer of on the left panel here, the stringing fibrous appearance inside the nerve cells, and this is known as neurofibrillary tangles. And today we know that these tangles are made up of highly phosphorylated and aggregated tau protein. So we also call it tau tangles. And on the right here, especially this structure in the yellow color, they are the amyloid plaques, the famous amyloid plaques. And these plaques are usually extracellular, and they are made up of the small beta amyloid peptides. So these are the defining pathological features of Alzheimer's disease. And the disease really preferentially affects older people, and it takes a long time to develop. In any given individual patient, even in the stage when the patient appear to be cognitively normal, their brain may already start to develop certain pathologies, especially start to accumulate beta amyloid and amyloid plaques. And then in terms of clinical presentation, it's very, very progressive and very gradual. And by the time individuals show memory deficits, this could already present very severe degeneration in the brain, including tau tangle formation, accumulation, as well as loss of nerve cells, neuroinflammation, and brain atrophy, and so on. So it can easily take 1 to 2 decades for the disease to manifest. And one thing that's really worrisome is the fact that since the disease preferentially affects older generation of people, and with our current aging society, the number of people living with Alzheimer's disease is going to increase exponentially. So today, in 2027, in 2026, in the United States, there are about 7.2 million people living with Alzheimer's. And as you can see, with life expectancy continue to rise, the number of people affected by Alzheimer's disease is expected to increase by a lot. So you may ask me, how do people get Alzheimer's disease? And it turned out that the disease etiology is very complex and is multifactorial. Genetics definitely plays a major role. So the disease usually runs in the family. But other factors such as sex, so Alzheimer's disease preferentially affects women. So the number of women affected by the disease compared to men is about 2 to 1. But environment, lifestyle, education level, they can all affect the risk of disease onset and the incidence of the disease. So given that so many different factors can contribute to the disease, how do we tease out how each different factor affects different type of brain cells and affect different aspects of the disease pathology or symptoms? So this is when we decided to use single-cell genomic approach to really get at the fingerprint of each individual brain cells from Alzheimer's disease brain samples. So we started this effort more than a decade ago, and the work is truly a close collaboration with my wonderful colleague at MIT, Professor Manolis Kellis at Computer Science and Artificial Intelligence, and also Dr. David Bennett, who is the director of the Rush Medical Center, and provided us hundreds and close to 1,000 of these precious post-mortem human brain samples for us to work with. And I also want to thank 2 former trainees, Hans Reddy Macy's, a former postdoctoral fellow in the lab, and Carlos Boy, a former graduate student in Manolis' lab. And together, we published more than 10 research articles. These are just some examples where we analyze different cohorts of humans. And our first publication back in 2019, we used single-cell transcriptomic analysis to analyze a total of 48 postmortem brain samples. And 2 years ago, we published another paper. Now we analyze more than 400 individual brain samples, and we also further extended the analysis to include multiple different brain regions and use different levels of analysis. And now I can say that we're sitting on single-cell genomics information of more than 800 individuals. So we're really sitting on a gold mine. Because these individuals from Dr. Bennett's collection, they also have very good detailed documentation of each individual's lifestyle, their medication history, their brain pathology, their imaging data, and many other important information. So we are in a very good position to analyze how each individual Alzheimer's disease risk gene affect the function and pathology of different brain cell types and associated with what pathology, as well as how lifestyle environments, such as how, in women, how hormonal replacement therapy medications would affect the disease progression and risk, as well as people's life experience, early life stress, social isolation, and many other factors. How do they affect the risk of developing Alzheimer's disease? And one thing I can say after analyzing more than 800 human brain samples is that we definitely now are very confident in the fundamental cellular processes that are severely affected by Alzheimer's disease. And the cellular processes, some of them are not surprising, such as synaptic signaling, genes and mitochondrial function, mRNA metabolism and processing. But one category that I think is extremely key and fundamental to Alzheimer's disease is lipid and cholesterol metabolism. How so? I'm going to tell you a little bit about that. So we know that there is a very major gene that is a very significant Alzheimer's disease risk gene. And this gene is known as APOE or apolipoprotein E. So in human population, this gene has 3 different versions. They are known as ApoE2, ApoE3, and ApoE4. And the 3 different versions differ from each other by 1 amino acid residue, such as the ApoE4 version has arginine, which are positively charged residue. And this ApoE4 version of the gene is the Alzheimer's risk gene. So, here I'm just giving you a very famous celebrity who came out to tell everyone that he has 2 copies of this Alzheimer's risk gene. And here is just to give you an idea how big an impact having this ApoE4 allele is when it comes to developing Alzheimer's. So if someone has no ApoE4 gene, then this person only has about 10% chance of developing Alzheimer's. And for those who carry 1 copy of ApoE4 gene, then the risk increased by 2 to 4-fold. And those who happen to have 2 copies of the ApoE4 gene, the risk increased by 10 to 15-fold. Pretty much that there's a very good chance this person will eventually develop Alzheimer's disease. So this ApoE4 version of the gene, as I said, carry this positively charged amino acid residues in these 2 positions, and is very likely to affect the conformation and folding of the gene product. So what is the consequence? So this apolipoprotein E or ApoE, the protein product normally is secreted outside the cells. And in the brain, it is mainly produced by the cell type known as astrocytes. And this ApoE product, its known role is to bind to cholesterol and other lipid species produced by the astrocytes and carry these different lipid species to other cell types to support their function. For instance, our nerve cells, neurons, they really need a lot of the cholesterol and lipids because they don't normally make this lipid by themselves. So cholesterol and different lipids are very important to support neuronal function. And also, cholesterol is the major component make up the myelin that protects the nerve fibers. However, while this ApoE2 and 3 versions of the gene product is very good at carrying lipid in these lipoprotein particles, ApoE4 has very compromised capacity to bind to lipid and cholesterol. So it has a very poor ability to, first of all, get the lipids out of the cell that produces lipids, and secondly, to bring these lipids to support the other cells who need the lipids. Okay, so the consequences is a massive defects in correct distribution of lipid and cholesterol species. So give you an example. Here in the ApoE4 microglia cells, you can see this lipid shown in the yellow color here. Now lipid just fill up the cells, okay, because it cannot get out. Similarly, in the ApoE4 astrocytes, these black dots also represent lipids, so they just accumulate in the cells. This is highly abnormal, okay? And this accumulation of lipid eventually impair the function of the cells and turn on immune response. And furthermore, here I'm showing you the myelinated axon fibers in a ApoE3 carrier person. So you can see their fibers are very well protected in this by this myelin sheath. But in the ApoE4 carriers, you can see this myelin is highly depleted, rendering these nerve fibers highly vulnerable to other damages. So, so this lipid accumulation turnout has already been described by Dr. Alois Alzheimer in 1907, where he, this is a translation from German, extraordinarily strong accumulation of lipid material in the entire central nervous system in Alzheimer's disease brain. So we think that this abnormal lipid distribution plays a key role in fundamental role in triggering a lot of the cascades of events, downstream events, leading to the development of other pathology and symptoms. So in my lab, we have been trying really, really hard to figure out how to counter these problems imposed by ApoE4. So we go through cell type by cell type in astrocytes, microglia, oligodendrocytes, brain endothelial cells, and neurons, and to characterize each cell type in the ApoE4 condition, how these different lipids accumulate and different pathology develop, and how we can actually identify small molecule drugs that can correct this ApoE4 imposed pathology and abnormalities in these different cell types. And we have also done non-biased high throughput screenings and whole genome CRISPR screening to identify protective factors. And so that's why I and other colleagues started a new company, new biotech company, to really try to translate these discoveries into drugs that hopefully one day can help people. So when it comes to available treatment for Alzheimer's disease, so we know that for 2 decades, there were only 2 kinds of drugs that physicians can prescribe to patients. And they are known as the choline esterase inhibitors to increase levels of the neurotransmitter acetylcholine in the brain. I guess the most famous drug is the donepezil, or known as Aricept, that is prescribed to almost all the patients. And then later on, another drug known as Namenda, which is AMDA receptor antagonist, that is prescribed to moderate to severe stage of the disease. And then in recent years, additional therapies have been approved by the FDA. This include the amyloid immunotherapy, such as Kisunla and Leqembi. And these drugs can tremendously reduce amyloid, beta amyloid plaques in the brain, even though the impact on the function and cognitive decline is modest. And I think one factor that makes some people kind of really reluctant to get on the drug is that this drug has side effects to cause brain hemorrhage or edema. And these effects are particularly severe in people who carry the ApoE4 risk gene. So obviously, other intervention methods are needed. And so my lab has described a very different approach, an approach that's extremely non-invasive and extremely safe to holistically changes the state of the brain. So this is a non-invasive brain stimulation approach. This approach really leverage on an intrinsic property of our brain, which is the neurons, they can fire synchronously. Connecting neurons, when they fire synchronously, they create brain oscillations, or known as brain rhythms or brain waves. And it turned out humans, as well as many animals, they can produce many different classes of brain rhythms or brain waves. And here I'm just listing some of the major classes that have been very well described in the literature. And the different classes of brain waves or oscillations differ from each other by frequencies, such as the delta wave, which is very, very slow, 1 to 4 hertz. They are most active during sleep. And today I'm really going to focus on the gamma waves, which are much higher frequency, ranging between 30 to 80 hertz. And these gamma waves are important and particularly strong during the function of attention or memory formation, decision making, and so on. So it's not surprising that in Alzheimer's disease patients and in animal models, the gamma rhythm is very much affected, okay, compared to normal healthy individuals. So now, more than 10 years ago, we figure out a way to boost the gamma rhythms or gamma waves in the brain. And we actually came up with a very non-invasive way to boost the gamma rhythms. So basically, we present initially mouse models of Alzheimer's disease with this patterned light and sound stimulation presented at the gamma frequency. We usually use 40 hertz light flicker and sound clicks. And this turned out to be extremely powerful in boosting the gamma waves in the brain. So we call this approach gamma entrainment using sensory stimuli or GENUS. So in 2016, a graduate student, Hunter Alcarino, first made the observation that with just 1 hour of gamma light stimulation, the brain of the Alzheimer's disease model mice undergo extremely dramatic transformation. For instance, these microglia cells, which are, normally the brain's immune cells, but they also play a role in phagocytosing toxic waste material. They show that after 40 hertz light flicker, you can see that the microglia look very different. Their cell body seems to be enlarged, and their processes are much more complex, indicative of a much more active microglia cells. And that's not the only consequence we see. We also saw that with GENUS stimulation, the amyloid plaques in the brain are also very significantly reduced. So if you compare this two hemisphere of mouse brain before and after gene stimulation, you can see reduction of the density of the amyloid plaques in multiple brain regions. So since our initial publication in the last 10 years, there have been hundreds and hundreds of publications from not just my own laboratory, but from many other groups throughout the world showing beneficial effects of genus of 40 hertz light and sound stimulation. People show that it can reduce inflammation, increase neuronal survival, and improve brain volumes, increase adult neurogenesis, improve cognitive function, improve axonal myelination, and reduce Alzheimer's disease, amyloid, and tau tangle pathology. And all of these beneficial effects happen when there are tremendous response from the different brain cell types, including microglia, neurons, the vasculature, oligodendrocytes, and so on. So, several years later, we finally start to understand why this very non-invasive brain stimulation approach can be so beneficial to the brain. So I just want to present to you one particular mechanism we identified. So this mechanism is known as the brainwave clearance or lymphatic clearance mechanism. So this involves the cerebrospinal fluid or CSF normally present in the brain's cavity or ventricles, but the CSF can enter the brain through the pumping of the arteries. So the artery pump the CSF into the brain, and the CSF can undergo this convective flow mechanism to clear the metabolic waste or the dirt from the brain. So you can imagine this. This is like the car wash, so the CSF flows through the brain like a car wash to remove the toxic metabolism. So this is a very simple movie to illustrate this process. So the CSF labeled in green enter into the brain, and then throughout this process, remove the dirt. And I should also say that it has been very well characterized. This lymphatic clearance mechanism is particularly active during sleep. However, a former graduate student, Mitch Murdoch, realized that this 40 hertz stimulation, even when animals are awake, he can see that the CSF is much more present in the brain, and this lymphatic clearance can be activated again during the awake phase. So, and this again is mediated through the pumping of the artery. So we can see that the artery becomes much more activated with the 40 hertz genus stimulation. So to summarize this whole body of work, so we found that after we provide the genus stimulation, there is increased gamma power, gamma wave, and then a special type of interneurons release a peptide. The peptide can then bind to the receptors on the blood vessel cells, and this transduces signals to signal the CSF to come into the brain, pump through the brain, and in the process, remove the amyloid plaques. So, with this very safe and non-invasive approach, the clinical trial has been conducted both at MIT and at the MIT spin-off company, Cognito Therapeutics. And this just to give you an idea how at MIT, we provide stimulation to the human subject with both light and sound. And in a previous publication, trainees Diane Chen, Brennan Jackson, and Ho-Jin Suk, we showed that this GENA stimulation, daily stimulation for 1 hour per day for 3 months, this leads to much reduced brain atrophy in human patients, and this is also associated with improved brain processing speed and visual memory function in treated patients. And I should also mention that Cognito Therapeutics conducted a phase 2 human study using Janus, and they reported a 76% lower decline in memory function and 77% lower decline in daily activity, ability of patients to carry out their daily life. And also there is a 69% reduction in brain volume loss. And so right now, Cognito Therapeutics is conducting a phase 3 pivotal trial they call the HOPE trial. And by recruiting 673 mild to moderate Alzheimer's disease patients. The trial is for 1 hour daily stimulation for 12 months. And later this year, we should learn something about the HOPE trial. So with that, I just want to say that since our initial publication in 2006 using this non-invasive stimulation, today there are more than 140 human trials throughout the world using 40 hertz stimulation for not just Alzheimer's disease, but many different diseases. So with that, I would like to thank all the people contributing to the work, dozens and dozens of lab members, both current and former lab members, as well as wonderful collaborators. Without their very important contribution, this work is not possible. And finally, I would like to thank all the funding provided by the NIH, as well as other foundations, as well as your support to make this work possible. So now I would like to end my presentations, and then I'm now delighted to welcome today's moderator to join me, Pam Belloc from the New York Times. Thank you very much.
Thank you so much, Professor Tsai, thanks to the audience. It's really a joy to be here with you. This is such a fascinating and important subject, and Professor Tsai, you covered a tremendous amount of ground, from the discovery of Alzheimer's to how it operates in the brain to potential solutions. A testament to the amount of interest this subject provokes. We have a number of audience questions, and I'm going to try to get to as many as I can. I'm fascinated by the Janus work that you were discussing. I guess I just wanted to start off because there's a lot of interest in potential therapies. How do you see the value of the 40 hertz light and sound intervention stacking up against these medications that have been recently approved and others that you see sort of coming down, coming through the pipeline? Can you sort of just give us a little overview of where you see the therapeutic situation now?
Yeah. So first of all, I think the biggest feature of Janus 40 hertz stimulation, as I said, is the extremely non-invasive nature. People don't have to take any injections or any procedures to receive this therapy, and it's also extremely accessible. All of the study subjects, we provide them with the device. They set it up at home. And they can just turn this device on every day for an hour to receive this treatment. So I think that compared to a lot of these other therapies, such as amyloid immunotherapy, I think patients have to travel to the hospital to receive the infusion every other week or every month. And then they are, very well documented potential side effects that physicians or neurologists have to monitor very diligently. So I think that so they are really, we're talking about very, very different therapeutic approaches.
There's a lot of interest in some follow-up questions on 40 hertz. So let me just quickly ask a couple, and then we'll move on to something else. But somebody was asking whether passive viewing of an approximate 40 hertz frequency on, say, their computer screen or their TV screen or something like that, would that be beneficial? And then there's another question about whether a similar frequency has an impact on bone density and balance. If there's other things that if mice or people could stand on a vibration plate and get the same frequency.
Yeah, very good questions. So on one of my slides, I think I went through it too fast because I was running out of time. The MIT device, which is a light panel, and we actually have a tablet in the middle or an iPhone or phone, your phone in the middle. So you can actually receive the light while you're watching a video or a movie of your choice. So definitely peripheral vision is enough. We actually performed EEG recordings and we didn't see much difference between you just staring directly or it's through your peripheral vision. We observe similar level of entrainment. So that's one thing. I think that definitely make the treatment much less painful. You can still do your things and receive the treatment. And then secondly, the second question is about vibrotactile stimulation. Yeah, I didn't have time to mention that we actually also published a scientific article on vibrotactile stimulation in mice. So we actually trained the mice to get used to this whole body vibration. And they got used to it, and then they received this whole body vibration 1 hour per day. And we found that it actually has additional benefit in terms of improving motor function in a mouse model that show motor deficits. So yes, and also in humans now we're trying to also do a trial, and we found that the vibrotactile stimulation is actually very soothing. It's almost like a massage. So I think that will become a very nice modality to be integrated into the GENES treatment.
Okay, thank you. There's a lot of interest in these anti-amyloid drugs, as you mentioned, and someone is asking, in light of the fact that there are these potentially serious side effects, what is the current thinking on the amyloid hypothesis for Alzheimer's? And maybe you want to explain quickly what the amyloid hypothesis is for people.
Yeah,
and what your thoughts are.
Yeah, so the amyloid hypothesis basically posit that the increased amyloid is the culprit that leads to a lot of this pathology and symptom, basically causing Alzheimer's disease. And there is a lot of genetic data supporting that, especially looking at the so-called early onset familial Alzheimer's disease. The genetic mutations seem to particularly affect protein products that are, part of the amyloid or amyloid production. And looking at so far the amyloid immunotherapy, when you completely get rid of amyloid, you can slow the disease by, on average 30%, 35%, which, I mean, is very significant effect. Although, it can be considered modest. So I have no doubt that amyloid definitely plays a role in the big picture of the disease. But the fact that after you completely get rid of amyloid, you still don't really completely stop the disease or cure the disease. Clearly, there are many other things that contribute to the disease. So amyloid is, in my opinion, unlikely to be the only factor.
Okay, great. There's a question here about the value of early detection and how close we are to being. I mean, I think we're very close to being able to sort of identify people who are at risk. We can already, right, with blood tests and genetic testing and things like that. But there's preventive treatments and preventive trials are already underway, some with the drugs that we were just talking about, some with other mechanisms. So can you sort of talk about, I mean, obviously that's sort of the holy grail here is to prevent rather than have to treat once people develop symptoms. Where do you think we are with prevention and how far out?
Yeah, so actually there are a lot of trials, I think, that can be considered kind of prevention trial. Basically changing lifestyle, right? Pam, you probably are familiar. There exercise and healthy diet kind of trial. And I think the most recent, I don't remember what it's called, but it actually significantly kind of delayed the disease onset. So clearly, I think that once one develops symptoms already, the brain already sustains tremendous damages, not just the pathology, but loss of neurons, loss of synapses, loss of connections. So to repair is difficult, and let alone completely restore the structure and function. So prevention is always a very good choice. Um, and and other than the lifestyle approach of prevention, I guess if one is thinking about using drugs, that then put a drug in the safety has to be very high bar, right? Because if you imagine someone to take this drug for the rest of their life since they're young, it just has to be something like a vitamin that's just completely harmless. So I don't think right now we have such kind of treatment yet. Um, and in terms of genus, we are conducting a so-called prevention trial. We are identifying people who have family history of Alzheimer's disease, but they are still cognitively normal. And so we are recruiting these people and provide them with the treatment for 1 year. And mainly we are going to just follow their biomarkers, such as their amyloid and tau level, to see whether this treatment can reduce the pathology and therefore delay the onset of the disease. And the trial is currently ongoing.
Yeah, fascinating. There are a few questions you mentioned, sort of lifestyle changes. And I see one question here about sleep and whether deep sleep, this is phrased, can it help wash out the brain junk? Is that true?
That's precisely what I show you, right? The lymphatic clearance that is most active during the non-REM sleep, which is the deep sleep. And that's when your brain gets cleaned. It's like your daily, car wash of the brain.
And then somebody else was asking about cholesterol levels. Is there a sort of optimal cholesterol level, a safe cholesterol level for brain health?
Yeah, see, I think I look at cholesterol as something that's, it has more to do with whether cholesterol is in the right place than the absolute level. Okay, because cholesterol is very important. As I said, it makes up all our biological membranes. So all our cells are basically made up of membranes. We have a cell membrane, we have organelle membrane, we have mitochondrial membrane, endosomes, all kinds of membranes. So we really need a tremendous amount of cholesterol and phospholipids and neutral lipids to sustain the structure of the cells and proper function of the cell. But if cholesterol is not in the right place, then there is a lot of problem. So I think cholesterol is important, but it has to be in the right place. If it's in the wrong place, then it's very damaging.
Somebody has a very specific question about microglial cells, and you are definitely the person to answer that kind of very biological question. It says the microglial cell appears to have two presentations as a result of the ApoE4 gene. One is the lipid accumulation and the other is the amyloid secretion. Are these related, or do they occur in different subsets of Alzheimer's?
Microglia definitely can accumulate cholesterol and other kind of neutral lipids. And this lipid accumulation from work in my lab and also now from many other people is strictly correlated with the inflammatory response in the microglia. So when there's lipid accumulation, cholesterol accumulation, this microglia then become much more inflammatory and they secrete inflammatory cytokines and just make the microenvironment more toxic. So I think that they're definitely probably same population of microglia. They accumulate lipid, they become inflammatory, they secrete toxic cytokines, and they cannot degrade amyloid and myelin debris and other brainwaves. So it's possible that through exosomes, this proteins get out of microglia again.
Okay, great. And sort of zooming in the other direction, this is kind of a broader question. I think it's a really good question with so much information available out there. Where can people go to find trusted sources, to find good resources, to help them understand, the discoveries and the scientific facts, and not get caught up in misinformation?
I think there are a lot of good sources. There's Alzheimer Association, which supports Alzheimer disease research and provides a lot of data on the disease. There's Cure Alzheimer's Fund, that also is a philanthropic organization that supports Alzheimer's research. And at MIT, we have the Aging Brain Initiative, and we also publish newsletters, a lot of newsletters that report the latest scientific research. So I think that there are definitely a lot of resource where people can learn about what's the latest.
Okay, great. And I think we have time for one more question. I'm going to kind of combine a couple because there's interest in I think the funding landscape and what happens, what are the potential consequences if federal funding dries up, and also sort of related to that, I think here somebody is asking how can alumni help move this research forward and sort of speed up the progress. So
Yeah, so the federal funding landscape has become very different and confusing um, um, to a large extent. Um, and um, and the federal government has changed the funding policy in terms of at the NIH. So it has become very challenging for, as far as I know, for scientists, not just at MIT, but basically scientists throughout the country. Everybody's having a hard time to get their grant funded. Um, and it is a difficult time. Um, and I want to say that my own experience, I think the one of the major factors that help me and my team to continue to make important discoveries and groundbreaking discoveries is really the research funding from philanthropy from you, because this money doesn't have a lot of string attached. We can use the money to really ask the most important question, not worry about the risk. And this money almost always leads to the most important discoveries. So I think it is more important than ever to support scientific research at this juncture.
well, I think we could talk for hours about this subject, but it's time to wrap it up. I want to thank you, Professor Tsai, for all of your insights and for the really important work that you and your colleagues are doing. I think if there's a takeaway here from today, it's that brain health obviously a very big issue. A lot of people being affected by declining brain health as they age, but there's promise with interesting and creative research like the projects that your lab is involved in. So there's sort of hope on the horizon, even though it may take a while. Thank you so much to the MIT Alumni Association for organizing today's session, and thank you to the MIT alumni community for joining and for asking such thoughtful and interesting questions.
Thank you. Thank you, everybody. Thank you, Pam.